In 2011, a team of scientists at Penn State University put an infected ant under a microscope and did something nobody had properly done before. They looked at the brain. What they expected to find was obvious. If a fungus is controlling an animal's behavior, overriding its instincts, redirecting its movements, making it climb to a specific location and bite down and die, it must be interfering with the brain. That is where behavior comes from. That is where control lives. The fungus had to be in there. The brain was untouched, not partially invaded, not damaged, not colonized, clean, intact, structurally normal. The fungal cells were everywhere else in the body, in the muscles, in the thorax, in the abdomen, forming dense networks, wrapping around muscle fibers. But the brain? The brain was fine. If the brain isn't being touched, what exactly is running this ant? To understand how strange this finding actually is, you have to appreciate what the ant does before it dies. This is not a simple behavior. It is not a single switch being flipped. Infected ant climbs. What the fungus produces is an elaborate multi-stage behavioral sequence. Abandon its colony and its pheromone trails, the chemical network ants normally follow with near-perfect discipline. Become disoriented and erratic, losing the navigational precision that defines ant movement. Shift from moving downward, ants naturally orient toward the forest floor where they forage, to moving upward, climb to a very specific height, typically between 25 and 30 centimeters above the forest floor, locate a specific surface, typically the underside of a leaf vein or plant stem. Bite down on the central vein of that leaf at a very specific angle, at a very specific time of day. Lock its jaw in a grip that is physically impossible to break. The mandibles fuse to the plant tissue through muscular force. Die in that position. That is not one behavior. That is a choreographed sequence of behaviors, each one leading to the next, each one precisely tuned to serve a specific fungal purpose. And the brain, the organ responsible in every other animal we know of for producing exactly this kind of complex multi-stage behavioral sequence, is untouched. The question is not just, how does Cordyceps control the ant? The question is, how does any organism produce complex behavior without touching the brain? Let's walk through what actually happens inside an ant's body after a Cordyceps spore lands on its cuticle. Not the broad strokes, the actual sequence. The spore adheres to the ant's outer shell through chemical bonding and begins to germinate within hours. Specialized structures called apressoria form, small attachment pads that generate mechanical pressure against the cuticle surface, essentially drilling in. Enzymes secreted by the fungus begin digesting the cuticle's chemical bonds. The spore breaches the barrier. Inside the ant's body, the fungus does something unexpected. Rather than immediately generating the filamentous structures, called hyphae, that fungi normally use to grow and spread, many Cordyceps species inside the host body remain in a yeast-like form, single cells that divide and multiply while floating in the hemolymph, the fluid that functions as the ant's blood. Yeast cells are harder for the immune system to identify and destroy than the hyphal form. The fungus is disguising itself. The infection remains largely invisible to the ant's immune defenses for an extended period. During this time, weeks, the ant is behaviorally completely normal. It forages, follows trails, interacts with nestmates, carries food. Nothing is wrong, nothing appears wrong, but the fungus is growing. At a certain threshold of cell population, a threshold we have not yet precisely identified, the fungus undergoes a transformation. The yeast-like cells convert into the hyphal form. Threads of fungal tissue begin extending through the ant's body cavity, reaching into muscle tissue, wrapping around muscle fibers, forming dense mats. It is around this transition point that the ant's behavior begins to change. The first change researchers notice is social. The infected ant begins to spend less time in the colony. Its integration into the colony's communication network, built entirely on pheromone signals, starts to degrade. It follows trails less reliably. It interacts with nestmates less frequently. One hypothesis: the fungus is already producing compounds that interfere with the ant's ability to produce or read pheromone signals. If the ant can't communicate effectively with its colony, it loses its social anchor. The chemical landscape that normally tells the ant where to go and what to do becomes noise. The second change is gravitational. Healthy ants have negative geotaxis. They tend to move downward, toward the forest floor. The infected ant begins to move upward, against gravity, against instinct, against 50 million years of evolution. The third change is directional. The climbing ant does not wander randomly. It moves toward the north side of vegetation in the northern hemisphere and south in the southern hemisphere. This is not coincidence. The north-facing side of leaves and stems tends to maintain specific humidity and temperature profiles, conditions better for fungal growth and spore dispersal than sun-baked surfaces. The fourth change is height-specific. The ant stops climbing at a very precise elevation. Field studies of Ophiocordyceps unilateralis infections in tropical forests have consistently found infected ants dying at heights of approximately 25 centimeters above the forest floor. Not 10, not a meter, 25 centimeters, with a consistency that is remarkable given that the ant has no ruler. Something is telling it when to stop. The fifth change is the bite itself. The ant clamps its mandibles around a leaf vein, not the leaf edge, not a random surface. The structural core of the leaf, the part that will hold the ant's body securely through the weeks of fungal development that follow. The bite is at a specific angle, oriented in a specific direction, and then the muscles lock. Within hours of the bite, the ant dies. The fungus has consumed enough of the ant's internal resources that the animal cannot survive. But the muscles remain locked. The body holds. The fruiting body, the stroma, emerges from the junction between the ant's head and thorax, growing upward, developing a capsule of spores at its tip, positioned precisely above the forest floor where other foraging ants will pass. Every step of this sequence serves the fungal reproductive strategy. Every step. David Hughes, a British parasitologist at Penn State, had spent years studying Cordyceps infections in Thai and Brazilian forests. In 2011, his team took infected ants at the moment of the death bite and sectioned their bodies for microscopy, cutting them into extremely thin slices and examining every tissue under the microscope. The fungal cells were densely concentrated in the muscles, particularly the muscles of the legs and the jaw. The muscles of the head were infiltrated most heavily. Fungal cells were found interspersed with muscle cells throughout the entire body. Critically, the fungal cells were not simply sitting in the spaces between muscle cells. They were physically connected to each other and to the muscle fibers themselves by structures the researchers termed hyphal bodies, dense masses of fungal cells connected by tube-like bridges. Hughes's team called these connections hyphal bridges. The brain, the ventral nerve cord, the insect equivalent of a spinal cord, and the major neural ganglia were all structurally intact. No fungal invasion, no mechanical disruption. The fungus appears to be generating movement directly in the muscles, independently of the nervous system. In a normal animal, movement works like this. The nervous system sends an electrical signal to a muscle. The muscle contracts. Movement results. The nervous system is the command structure. Without it, muscles don't move. What Cordyceps appears to do is insert itself not at the command level, but at the execution level. The fungal cells don't tell the brain to tell the muscles to move. They interact directly with the muscles themselves, producing chemical signals that trigger contractions independently. The ant's brain may still be issuing its own commands, but if the muscles are being chemically controlled by the fungus, the brain's commands become irrelevant. The fungus has not hacked the brain. It has made the brain irrelevant. If muscles are being controlled chemically, then somewhere in the fungal arsenal there must be specific compounds responsible. Finding those compounds has been the central challenge of Cordyceps research for the past 15 years. The difficulty is enormous. The infected ant's body contains thousands of chemical compounds from both the ant and the fungus, many of them present in vanishingly small quantities. Mass spectrometry, a technique that identifies molecules by their mass and structure, has been the primary tool, and slowly, piece by piece, the chemical picture has begun to emerge. Guanidinobutyric acid, GBA, is one of the most significant finds. The fungus produces this compound in notable quantities during infection. In other biological contexts, GBA is known to provoke seizure-like activity in muscle tissue, intense, repetitive muscular contractions. This is almost certainly what produces the death grip. The mandibular muscles of infected ants contract with such sustained, overwhelming force that the jaw physically fuses to the plant surface through muscular pressure alone. This is not rigor mortis. This is an active, chemically driven contraction occurring while the ant is still alive. Sphingomyelin pathway compounds have also been identified. Sphingomyelins are lipid molecules, fat-based signaling compounds, that play a role in how muscle cells respond to or ignore incoming signals. Manipulation of sphingomyelin pathways could make muscle cells more susceptible to the chemical commands being issued by the fungus, effectively amplifying its control. Biogenic amine modulators have been found as well. In insects, biogenic amines like octopamine and serotonin regulate fundamental behaviors: movement, aggression, foraging drive, gravitational orientation. These are the insect equivalents of our own dopamine and serotonin. Manipulating these compounds could explain many of the early behavioral changes: the social withdrawal, the loss of normal orientation, the shift from downward to upward movement. The full picture is almost certainly more complex than any single compound. Cordyceps has evolved a coordinated chemical symphony, different compounds triggering different behavioral shifts at different stages of the infection. The early social disengagement, the directional reorientation, the climbing, the height-specific stopping, the bite, the lock. These may each correspond to specific chemical signals being deployed by the fungus at specific points in the infection timeline. A sequential pharmacopoeia, deployed in order to produce a result. We have not yet mapped it completely. Researchers believe there are significant compounds still to be identified. The fungus is, in a biochemical sense, more sophisticated than our current tools can fully characterize. Here is something that almost never gets mentioned, and it should. The fungus does not simply invade the ant and destroy it from the inside. In the process of colonizing the ant's body, it displays what can only be described as selective restraint. Analysis of infected ants has shown that the fungus carefully avoids the ant's vital organs during the early and middle phases of infection. The heart, the digestive system, the major muscles required for locomotion, these are preserved. The ant remains physiologically functional right up until the moment of the death bite. This is not accidental. An ant that dies too early is useless to the fungus. It needs the ant to walk, climb, orient, and bite. A dead ant cannot climb. A dead ant cannot grip a leaf vein and hold on. The fungus farms the ant. It consumes non-essential tissues while preserving essential ones, maintaining its vehicle in working order precisely long enough to deliver itself to the optimal position. This level of precision, selectively preserving specific tissues while exploiting others, maintaining an organism's function while redirecting its behavior, implies a degree of awareness in a purely biochemical sense about what the host needs to remain useful. The fungus has no consciousness. It plans nothing. But natural selection has produced something that functions as if it does. The death bite happens at a very specific time of day. Field researchers studying Cordyceps infections in tropical forests have found, repeatedly, that infected ants perform the death bite near solar noon, the point in the day when the sun is at its highest. In the forest understory, solar noon corresponds to specific microclimatic conditions, temperatures at their daily peak, humidity at a relative low point, airflow patterns shifting as the day's convection cycles reach maximum. The spores released by the Cordyceps fruiting body fall downward into the airspace below. At solar noon, the air currents and temperature gradients in the forest understory may be optimal, carrying spores further, keeping them airborne longer, depositing them where ant traffic peaks. The fungus is scheduling its own reproduction around the physics of the forest. How? The most likely mechanism involves the ant's own circadian biology. Insects have internal biochemical clocks that regulate daily cycles of activity. The fungus may produce compounds that intercept the ant's circadian signaling, triggering the death bite when the ant's own internal clock signals that it is midday. Alternatively, the fungus itself may have light-sensitive biochemical systems, receptors that respond to changes in ambient light intensity and trigger the compound releases that produce the death bite when light levels peak. Either way, a fungus with no nervous system, no eyes, and no brain is synchronizing a behavior to a specific time of day, in a specific forest, at a specific elevation above the ground, with compass orientation factored in. If a human engineer described this as a designed system, we would call it extraordinary. In nature, we call it evolution. The Penn State finding about the intact brain opens a question that researchers have generally been cautious to ask directly, but that is genuinely difficult to dismiss. At the moment of the death bite, the ant's brain is intact. Its neural activity has presumably not been disrupted. There is no structural reason for it to have ceased. The ant may, in some sense, be conscious, experiencing something, as its muscles carry it to the leaf, as its jaw locks down, as it waits. We are accustomed to models of behavioral control in which consciousness is disrupted along with behavior. Drugs alter how people feel while altering what they do. Brain injuries change both experience and action. The Cordyceps model raises a different possibility. An organism whose brain is structurally normal, whose neural activity may be continuing, whose inner experience, if there is one, may be entirely intact, while its body does something its brain did not choose. What is it like to be an ant whose muscles are moving without you? We do not know. The ant cannot tell us, but the question should not be dismissed. The Cordyceps case is, in this sense, philosophically remarkable. It is a natural experiment in the separation of consciousness from behavior, in the possibility that what a body does and what a mind experiences can be decoupled by a sufficiently sophisticated chemical intervention. Neuroscientists studying the relationship between intention and action have long been interested in cases where the two diverge. Cordyceps provides the most extreme natural example. Ophiocordyceps unilateralis is the most studied, but it is far from the only behavioral manipulator in the Cordyceps family. Different species produce radically different manipulations in different hosts, and the variety is astonishing. Cordyceps species targeting moths and caterpillars produce a different manipulation entirely. The infected caterpillar climbs to the top of a plant or tree and clings there, but it does not bite. It dies in place, body hanging from the vegetation, positioned to rain spores down on the plant tissue below where other caterpillars feed. No death bite needed. Gravity and body weight do the work. Massospora cicadina, a fungus that infects cicadas, produces perhaps the most disturbing behavioral manipulation of all. The fungus replaces the abdomen of the infected cicada with a mass of spores. The cicada continues to live and move, physically missing its entire abdomen. Males infected with the fungus continue to attempt mating. The fungus has been found to produce compounds that cause infected males to mimic the wing flick signals of females, attracting healthy males. The infected male then transfers spores during the attempted mating. The fungus has co-opted the cicada's reproductive behavior to spread the infection. Massospora also produces psychoactive compounds, cathinone, related to amphetamines, and psilocybin. The cicada may literally be high as its body is consumed, the compounds potentially keeping it active and mobile far longer than it would otherwise survive, maximizing spore dispersal. Each of these strategies is different. Each is precisely adapted to its host's biology, ecology, and behavior. Each has been sculpted by evolutionary time into a system of manipulation that would be almost unbelievable if encountered only in a textbook. The question, how does Cordyceps control its victims, does not have one answer. It has as many answers as there are species, each one a different solution to the same problem, each one a different masterwork of evolutionary engineering. Richard Dawkins, in his 1982 book, The Extended Phenotype, proposed an idea that organisms like Cordyceps make concrete, that genetic influence does not stop at the boundary of an organism's body. Dawkins' concept was this: genes express themselves through the bodies of the organisms that carry them, but the effects of those genes extend outward, into the behavior of other organisms, into the structures those organisms build, into the ecological systems they shape. A beaver's genes are expressed not just in the beaver's body, but in the dam the beaver builds and the pond that dam creates. The dam is part of the beaver's extended phenotype. Cordyceps takes this to a radical extreme. The fungus's genes are expressed not just in the fungal body, but in the ant's muscles, the ant's position in space, the height of the bite, the compass orientation, the time of day. The dead ant is part of the Cordyceps genome's expression. The ant, in this framework, is simply one organ among several in the fungus's extended body, a limb, a vehicle, a tool shaped by evolutionary selection over millions of years to perform one specific function. This reframing does something important to our understanding of organisms and their boundaries. We tend to think of an organism as a discrete, bounded thing, inside which the organism exists, outside which it does not. Cordyceps blurs that boundary into something almost incoherent. The fungus's biology extends seamlessly into the ant's biology, into the ant's behavior, into the ant's precise location in the forest. Where does the fungus end? Where does the ant begin? Cordyceps sits at the intersection of neuroscience, mycology, ecology, evolutionary biology, and the philosophy of mind, and it keeps producing results that challenge assumptions. It challenges our assumption that behavior is produced by brains, showing that muscular systems can be controlled independently of neural command. It challenges our assumption that a parasite is a simple relationship, one organism taking from another. The cordyceps relationship with its host is more intimate, more coextensive, and more technically sophisticated than any simple taking. It challenges our assumption that evolution can only produce incremental change. The level of precision required to synchronize spore release with solar noon, to position a dead ant at 25 centimeters with compass orientation, to chemically sequence 6 distinct behavioral phases across 3 weeks of infection. This precision was not designed. It emerged from variation, from selection, over 48 million years, without purpose or foresight. We did not design systems this precise. We are only beginning to reverse engineer them, and every time we look more closely, there is more to find. We started with a single finding in a microscope lab, an ant doing everything a cordyceps ant does, and a brain showing none of the damage anyone expected. That finding opened a question that is still not fully answered. How does cordyceps control its victims? The answer, as we now understand it, not through the brain, through the muscles, directly, through a chemical arsenal, some compounds identified, many still unknown, that produce movement, orientation, and behavior without neural input, through a sequence of manipulations calibrated to the host's biology and the forest's physics across 6 distinct behavioral phases, spanning 3 weeks. And the deeper answer, cordyceps reveals that control is not as simple as we thought. That behavior is not only what the brain makes, that the boundary between organisms is more permeable than we imagine, that a fungus with no nervous system, no eyes, no brain, can produce in another animal a behavioral sequence more precisely calibrated than anything most animals achieve voluntarily. The ant climbs, the ant bites, the ant dies. And somewhere in the forest, another spore falls. Subscribe and explore the hidden kingdom. Something stranger is always waiting around the next corner.